Connie Lynn Rogers.
And I am Ty Parks.
And we are proudly representing the Georgia FFA Association.
Our research indicated that feeding hogs medicated feed,
like this,
does not increase the antibiotic resistance of bacterial colony forming units. In the feces of the pigs, we use skills and information learned from a statistical analysis and repeated trials to support a hypothesis. For example, we applied steps from soil bacteria experiments to culture bacteria in our animal systems research. We also use a scientific method and collaboration with experts as a team to solve an agricultural problem.
Question 1. I have been showing swine for 11 years, and my dad is the owner of a large hog farm in Onega, Georgia. Many farmers like my dad give swine and other animals medicated feed such as this. One day I was scrolling through Facebook and stumbled upon an article indicating that giving animals medicated feed was harmful for the environment because of the chance of increased antibiotic resistance. I wanted to dig deeper into this subject.
Having identified the problem, we began using the scientific method to further investigate the issue. We then conducted a review of literature where we gathered information that helped us form our hypothesis. The hypothesis stated that there would be a statistical similarity in the number of antibiotic-resistant bacteria found in the feces from the hogs fed with medicated feed and hogs fed with the non-medicated feed, with a critical value of 0.95.
Question 2. Because of our project, we were able to solve a problem for farmers using the scientific method. My partner and I both learned more information about bacterial antibiotic resistance. We also learned about the withdrawal periods for antibiotics with information in our review of literature. We were able to practice skills such as laboratory safety and the ability to perform precise measurements on recording bacterial colonies.
This led us to learn how to perform a statistical analysis and to evaluate our results on a larger scale. We used an online website where we entered our data into a t-test calculator. This generated statistical information such as our p-value of 0.96. Upon doing so, we developed a confidence interval to describe the outcome of the statistical analysis. Question 3. We collaborated with Dr. Marshall Bennett, a doctor of veterinary medicine.
We informed him about a previous study from researchers Montgomery and Gay, who investigated soil bacterial antibiotic resistance. Dr. Bennett stated that a concern for livestock farms was the outcome of antibiotic resistance caused by production practices. He also stated that these could possibly occur in animal feces following an antibiotic treatment or feeding of medicated feeds.
He advised that this would be an important issue to investigate. While researching, we found that the FDA proposed a ban on certain antibiotics and feeds without investigating the accuracy of this allegation. Dr. Bennett's input was valuable to the integrity of this project.
Question 4. We had several conclusions on this research project. In our first conclusion, our hypothesis was accepted, and we had a p-value of 0.96.
And our second conclusion was that the comparison group fed with non-medicated feed actually had a higher count of antibiotic-resistant colony-forming units than the experimental group.
However, we are most excited about our third conclusion on the financial impact on hog farmers. Shabandia indicated that there were 77.34 million pigs in the United States. We found that the butcher value of a market pig to be an average price of about $120 per market hog. We use this information to make a conservative estimate that medicated feed is worth more than $931 million to hog farmers in the United States.
Thank you for allowing us to share how we use the scientific method to find that feeding hogs medicated feed does not increase antibiotic-resistant fecal bacteria.